Hi, everyone. My name is Margot Helms, and I wanted to share with you my Capstone Project. The topic I have chosen for this project is how can new kinds of mRNA vaccines help cure cancer? So on the left, I have an image of RNA, which is ribonucleic acid, and we'll get more into this later, but as you can see, there's some of the nucleotide bases and the single strand of RNA as opposed to the double strand for DNA. So the question we're going to ask right now is, what is mRNA? mRNA stands for messenger RNA, but in order to answer and define this a little bit better, I need to backtrack a little bit. So going back to high school biology, as we can all remember, we know that the cell is the building block of life. Cells make up everything, from muscle tissue to heart tissue. Even hair follicles are made of cells originally. And so when we are referring to cells, I like to think of them as a machine. Machines always work together with all the little parts and cogs so that they can turn and perform a generalized function. And it's the same with cells. Cells have to work together with all their little cogs to perform a function. And these cogs are known as organelles. For this project specifically, one organelle I want to talk about is ribosomes. So ribosomes work really closely with mRNA. They work hand in hand, I guess you could say. Ribosomes work in protein synthesis, which is really the main key component of mRNA, right? So mRNA is ribonucleic acid, which functions as a messenger, hence the name messenger RNA. And it is composed of the 4 nucleotide bases, adenine, cytosine, uracil, and guanine. And mRNA is a direct copy of a portion of DNA's nucleotide bases. However, RNA and DNA are different in that DNA is the double strand, RNA is a single strand, and there is a different base in DNA that mRNA does not have. But DNA is really important to the cells, and it's stored in the nucleus of the cell, which it cannot leave from because that information is super important, so they don't want it to get damaged. So instead we said mRNA, and to copy the nucleotide bases sequence in its own little language as mRNA, and then it is translated later into what eventually becomes a protein when it goes through the ribosome in protein synthesis. So moving on, I wanted to talk about the history of mRNA vaccines. So mRNA was kind of originally discovered around 1960s, and it was just left as a discovery. No one really put too much energy or effort into it until the 1990s, which is when people realized that mRNA had a lot of importance to it, and it could be used to help solve issues like cancer. So in the 1990s, some scientists and researchers were dabbling in mRNA technology to try and help figure out ways to fight against cancer. But after that, it didn't gather a lot of attention from the medical community and was kind of pushed to the side until 2010, where the founders of Moderna actually found this mRNA technology super interesting and decided to start and study it more closely and bring it to the attention of the medical community. So mRNA was beginning to be worked with kind of more in 2010, and then 2020 was when the COVID-19 pandemic hit, as we all remember. And due to the pandemic, mRNA came to the forefront of medicine and was beginning to be used in order to vaccinate against COVID-19 with the COVID-19 vaccination, which uses mRNA technology. And then in the present day, because of the new growth in attention to mRNA vaccines, there's a bright future ahead of us. There's many different problems that we can try and tackle with utilizing mRNA. So how do mRNA vaccines work? The first step is making the synthetic mRNA. So this mRNA is made outside of the body in a lab, and then it's surrounded by liposomes, which is the key to sending in mRNA from outside the body without it being destroyed. Next, we get to the vaccination portion. So the vaccination is when the synthetic mRNA is actually inserted into the body. And then eventually, next step is it's absorbed by the cells. And once it is inside the cells, it can interact with the ribosome, and protein synthesis can happen. And then finally, after the protein production, once that protein's made, it can either help the cell by showing immune cells things that we want to target by the proteins that we made, or it can also prevent things from coding. For instance, like cancer cells, we want to stop tumor overgrowth. So the mRNA can help prevent that. Next, we want to look at the chemistry of the mRNA vaccines. So let's start with talking about the chemical structure of RNA. If we look in the right-hand corner, there's the molecular structure that I'm going to refer back to a couple times. One component of the molecular structure is the ribose sugar, which looks like a pentagon there, and it has 5 carbon and 1 oxygen. And these ribose sugars are kind of like the backbone / head of the RNA. There's always going to be these multiple ribose sugars because it's the only way for the nucleotide bases to attach. And then additionally, we have the phosphate groups, which is a polyatomic ion that we discussed in class a little bit. The phosphate group is also part of the backbone of RNA. It wouldn't be able to form or function without it. And then lastly, I want to talk about the nucleotide bases. The nucleotide bases are the adenine, guanine, cytosine, and uracil. And these are nitrogen based, hence the name nucleotide bases. And they have a lot of nitrogen, some oxygen in them. And if we look back at the top left corner at the image, you can see the little oxygons with the N is the nitrogenous bases. While we have those pentose sugars, the ribose sugars, and then the phosphate groups. So here's a better image of all of the nucleotide bases with the phosphate groups and attached to those ribose sugars. And if we also look closely, we can see an OH, which is a hydroxyl group. This is unique to RNA. There is no hydroxyl groups in DNA, and we will talk more about this later. I also have an image of how the vaccine for COVID was synthesized using mRNA information, providing immunity for later infections. So next we want to talk about the oxidation of RNA. So because of the hydroxyl group, that OH there, RNA is really easy to oxidize, which makes it very unstable, because if it gets a chance to interact with water, it's going to transfer molecules, hence oxidation, from the RNA structure to the water, creating new products. And this in turn cleaves the RNA, which makes it very unstable. It halves the message that we're trying to code for the protein in half, and it is almost better off without it being there at this point. So because of that, and because of the risk of oxidation due to the OH groups, we have invented liposomes, which is the covering that is surrounding the mRNA vaccines. So liposomes are amphiphilic molecules, meaning they both have a polar and non-polar portion to the molecule. The addition of the amniphilic layer prevents oxidation by creating kind of like a surrounding of that polar head, meaning it doesn't like water. And so it's rejecting the water, keeping it safe from having that oxidation interaction occurring. And this way it can get safely inside the cells, that we are trying to create a protein synthesis within. And then once it is inside the cell, we want to talk about the ionizable lipids. So this creates a polar molecule which helps infiltrate the body without the immune response, like we just said. And then once it's inside, the lipid's going to break away due to a lowering in pH. And this is because the cations of the lipid, which is the positively charged portion of the molecule, are attracted to the endogenous endosomal phospholipids, anions. So those negatively charged ions and positively charged are going to be attracted to each other because they want to get that full octet, and this breaks apart the lipids in order for the protein synthesis to take place. So how can mRNA help cure cancer? Well, the mRNAs are of particular importance. They are about 22 nucleotides long and function in gene regulation in most eukaryocytes. They can inhibit or silence gene expression by binding to target mRNA and inhibiting translation, thereby preventing functional proteins from being produced. Many miRNAs play significant roles in cancer and other diseases. For example, tumor suppressor and oligogenic miRNAs can regulate unique target genes, leading to tumorigenesis and tumoral progression. So I took this from author Yuan on the website of Britannica. Some important things I want to talk about from this quote is the mention of how mRNA can help suppress tumor growth. So if we give the body synthetic mRNA through mRNA vaccines, this will be a way to show the body what not to allow to generate for proteins or what to attack. So different ways mRNA fights cancer. The synthesized RNA and what it does against cancer. The RNA is shown to the body and becomes a way to identify a cell that is to be terminated. So like I said, injecting that RNA, that protein synthesis will take place, and from a new protein being shown to the body, it's going to interact with those immune cells, and the immune cells are going to become aware of an enemy per se. So in the case of cancer cells, some things they'll be able to identify are specific molecules that are typically found within tumors. That's what some of the synthetic RNA could technically be made to send the message about. Or they could also inform the cells how to stop unnecessary cell growth, which is what tumors are. So if we can find the cells that are growing and identify a way to tell the cells to stop growing them, then that will also help with tumor suppression. And then another point I want to make is that mRNA can help kickstart T cell production to attack individual patients' cancerous cells. So if the mRNA vaccine is a personal one, with specific genetic blueprints from an individual patient, then the T cells, which is an immune cell that helps kill cells, will be activated because of this protein synthesis that wasn't there before the vaccine. And another portion of using mRNA for cancer, you can do a specific patient's cancer vaccine, which is made from their material specifically, or it can be one that's targeting common molecules like we talked about in one of the points above. So here's my resources, and I want to thank you for your time, and I hope you have a great rest of your semester.